Morning, welcome to STEM Ambassadors Live, and I'm joined today by Bharath, and Bharath is a associate principal scientist at AstraZeneca, and he's going to speak to you about with his talk, A Globetrotter's Guide to Scientific Research, his research journey. So I'm going to hand over now to you, Bharath.
Thank you very much. Thank you. Thank you very much, Vanessa. As Vanessa kindly introduced, I'm going to talk about A Globetrotter's Guide to Scientific Research. And this talk will mainly focus on trying to enthuse the next generation of scientists into the wonders of science and the fact that as a scientist, you need to have a multifaceted personality rather than a very unique perspective on how you get things done. And that's precisely the kind of message that I'll be driving home during the course of this talk. And in case you have any questions, please reach out to Vanessa or to me in case we can help you with something. So this is me, as you can see, and this is 10 years back. And I did this painting in gouache and watercolors. And so just to highlight the fact that as a scientist, I have varied interests that kind of add to my scientific ability and acumen about thinking about scientific problems. And as Vanessa pointed out, I am an associate principal scientist with AstraZeneca, and that's my daytime job designation. And I'm a drug hunter, so I look for novel small molecules that can be used as a way of treating several diseased conditions. So getting back to my origins, as you can see here, I'm showing you a world map, and I would like to make sure to say that all these images have been borrowed from internet, and just for the sake of making sure that I kind of have a lot of colorful things on my slides, and it's exclusively for educational purposes, and it's nothing to do with AstraZeneca. So this is purely in my role as a STEM ambassador. So you will see that this is the map of the world, and I come from a very tiny island that is sandwiched between India and Sri Lanka, as is shown in the zoom right here. And this kind of creates a very unique ecosystem, because as you will see that Indian Ocean is here and the currents are quite strong. But what happens is as the currents come across and hit the mainland India, it's kind of blocked by this mass of Sri Lanka here. So the water here is kind of very tepid, very warm and very shallow and very mild. And it's kind of a very unique ecosystem called as the Gulf of Munnar Marine Biosphere Reserve right here. So you will see that a lot of tsunamis, there were instances of a lot of tsunamis in the previous couple of years, but none of them kind of did any damage here because of the very fact that Sri Lanka took the bulk of the brunt, likewise the eastern coast of India. But then this region was highly protected. And this region, because of this unique location, has a lot of unique traits and interesting aspects. For example, it's the home for manatees, a specific kind of manatees called dugongs. And as you all might know already, they feed their young ones using mammary glands. And they are not fish, they are mammals, in fact. And the island is connected to the mainland by a huge bridge, as is shown here. It's kind of very long. It takes approximately 15 minutes, if not more, for you to go from one end to another in a car driving, and it's kind of very scenic, as you can see. And the island is also home to one of the most beautiful temples in the whole world, dedicated to a Hindu god called Shiva, as is shown here. And it has the longest corridors of any temples for any building in the whole world. So it has a lot of unique things, quite beautiful place to come from. Subsequently, because of the fact that my father was employed in a paramilitary organization, I traveled across the length and breadth of the country, as is shown here. And you can see that it covers a lot of unique ecosystems. I'll walk you through this in a minute. So as I said, I belong to Rameshwaram right here. And as part of my travels, I have been to Jodhpur in Rajasthan, a state in India called Rajasthan, which is famous for its deserts, as is shown here. And then from there, I went to the northeast of India, a place called Silchar, which is very famous for its tea plantations and heavy rainfall. In fact, a place in the northeast called Cherrapunji receives the highest rainfalls in the whole world. So that's what the record was sometimes back. And subsequently, from here, we went to stay at a place called Sikkim, and you will see that it has this very scenic view of Mount Everest. It's not Mount Everest. In fact, it's Kanchenjunga. It's the second highest peak in the range of Himalayan peaks right here. So you could see the peak right from where you used to stay. So, and apart from that, of course, if you come down south, this is the kind of view that you see. So I had been kind of exposed to various geographies as I traveled the length and breadth of India during my formative years and during my schooling. And bulk of my schooling was done at a very prestigious institution called Kendriya Vidyalaya. So I'm showing you here the emblem. And subsequently, when I started my graduate studies, I went into some of the most prestigious universities that India can boast of. And the cream of cream is Indian Institute of Science, where I did my part of my PhD, and JNCASR. So this is where I kind of learned the rudiments of the skills that I'm employing currently as a drug hunter in my job. So with that brief introduction and having finished my PhD in India at Bangalore, I started on my global tours. So from India, I went all the way to the Atlanta, a place called Atlanta, which hosts a whole lot of universities, prestigious universities. Emory University is one of them, Georgia Institute of Technology is one of them, and Georgia State University is the other one. So I did my postdoctoral studies at Georgia Institute of Technology here, and I studied a problem at the interface of chemistry, medicinal chemistry, computational sciences, and enzymology. And that's precisely the kind of skills that you need as a drug hunter. And I'll kind of briefly take you on the journey to show how this kind of globe-trotting and learning a whole lot of different skills has helped me evolve as a drug hunter during the past 10 or so years. And of course, I spent some time at Harvard University as part of a collaborative project, and I was extensively funded and generously funded by a national body from Washington, DC, called as the National Institute of Health, to pursue all the studies. You keep upkeep. You need some money for your upkeep. So that's precisely where this angle comes from. And subsequently, having spent 5 or 6 years in Atlanta, I decided to fly to Lisbon to take up a position at Instituto Gulbenkian de Ciencia. It's in a very scenic suburb of Lisbon called Oeiras, and you could walk to River Tejo, and where the river is almost merging with the ocean. So it's a very beautiful place to stay at, and you can walk all the way to a place called Cascais, traversing Estoril, Carcavel Ocean, all those places, and then you go to Sintra, which is a kind of hilly region, completely covered in fog and mist, kind of very magical. So I studied there, and I studied a very unique problem where I tried understanding how our immune system kind of differentiates between our own nucleic acids and the nucleic acids of foreign particles like viruses, right? When virus infects you, how does your system know that it's a virus, and to make sure that it doesn't harm your own cells while still harming the virus, kind of. It's a very deep and intricate problem that I'll not go into details of, but of course you should be able to pick up literature on the internet on these things. So I was once again generously funded by associations like the Marie Curie Actions Fellowship, and as part of the European Union funding. And having kind of finished my training here, I decided to move to the United Kingdom, and I joined an industry, which was a sea change for me from the academic training that I had been exposed to. But thankfully for me, I realized how important it is for you to acquire skills and then kind of transplant it from one framework of thinking to another framework to kind of bring the value addition that comes with such a kind of transplantation. And then I joined AstraZeneca. And as I pointed out to you, my daytime job is an associate principal scientist at AstraZeneca. I'm sorry, the slides are somehow not behaving well. So yeah, so my daytime job is an enzymologist at the mechanistic and structural biology unit within AstraZeneca. And what I do here is I try looking at how small molecule drugs or potential drug-like molecules interact with their protein target of interest. How long does it take for the small molecule to load onto the enzyme of interest? How long does it take for it to dissociate from the enzyme of interest? How long does it stay on the enzyme of interest? These terms might seem very simple, but they have huge bearing on how a drug acts on the molecule target of interest and brings about the kind of beneficial effect that we would want drugs to have. So this is my principal daytime job. I do a whole lot of other voluntary. I'm involved in a whole lot of other voluntary roles, as is shown here. I'm a faculty with faculty opinions. I'm also an editor with British Journal of Pharmacology, and I'm editor with Current Opinion in Pharmacology, two of the best pharmacology journals out there. I'm also an editor with a biochemical journal called the FEBS, Federation of European Biochemical Society Journal, which is a very prestigious biochemical society journal. And so I'm an editor with it. I'm also an ambassador in the Biochemical Society, and I'm also an ambassador with British Pharmacological Society. So in all these varied roles, I make sure that I kind of translate the enthusiasm that I have for my science to a wider audience and make sure that they get equally enthused by pursuing careers in STEM. And so apart from these main roles and voluntary roles, I'm also involved in a whole lot of activities that kind of, I think, complement and enrich my skills. One is I do a lot of charcoal painting, and I was lucky that one of my charcoal panel of charcoal portraits got selected to be highlighted on the cover of FEBS journal. I do a lot of multimedia paintings, as you can see here, kind of very rich, and I'm trying to replicate here the stained glass theme, the colorations and all those things on Indian mythological themes, as you can clearly see. I use a lot of different pigments here just to make sure that I get the glow and the kind of coloration right. I also do a lot of e-art, and again, that's once again an example of an e-art of mine that got published on the cover of Journal of Cell Science is this, where I'm trying to depict a stress granule. I write quite extensively literary fiction, as is shown here. I have published a book of fiction called Formless Meanderings. I do a lot of sculpting too, and I'm very, very passionate, as you would have already understood, about science outreach and communication. I publish quite extensively on how to teach enzyme kinetics, what is the history behind enzyme kinetics, how to make enzyme kinetics more accessible to a wider audience so that they can appreciate the nuances of the subject. So these are some of the things that I do apart from my regular day job and volunteering activities associated with all the other things that I said. So what got me interested in science of enzymology? I'll depict here two examples that will kind of showcase as to how science is so exciting. So there was this ship called Titanic that sunk in the frigid oceans of Atlantic 100 years back, approximately. I mean, it was, I think, the year 2014 when it sunk in the waters, and as is shown schematically here. 100 years later, there was a deep sea excavation project that kind of retrieved this piece of paper from the hull of the sunken Titanic. And as you can see, you can clearly see the writings, quite legible, and the paper has not kind of disfigured with time. So that is because of the very fact that the fact that it has survived in the hull of Titanic for close to 100 years has a lot to tell you. And as anyone who is basically acquainted with how, what paper is would know that it's a polymer of glucose right here. Right? It's a slightly different polymer of glucose than, let's assume, sugar, but then it is a polymer of glucose. And the fact that a sugar polymer has survived in deep waters for close to 100 years talks a lot about why enzymes are important. Can you imagine if you eat your breakfast in the morning, essentially bread, which is nothing but starch, which is again polymer of glucose, and wait for 100 years for it to get digested, for you to get the necessary energy? Life will not be possible. So you need enzymes. And the fact that the paper has survived in the hull of Titanic was precisely because of the fact that there were no bacteria in the deep underwater frigid waters, and hence there were no enzymes that could degrade the piece of paper. And this I think kind of impresses upon what enzymes do. Another example is they kind of accelerate the rate of reactions for them to be compatible with life. So life is synonymous with enzymes. I'll tell you another example. This is a fictitious piece of picture that I took from a movie, a very popular movie called Jurassic Park, where the protagonist is peering into an ember which has trapped a mosquito inside it. So the mosquito inside, before it got trapped in the ember, had a blood meal on a dinosaur, right? And then subsequently it got trapped and was trapped in this ember for millions of years. What the protagonist did in this fictional narrative movie was to extract the mosquito from the ember, extract the dried blood from the gut of the died mosquito, and extracted the DNA of the dinosaur because it was dinosaur blood, dino blood. So it took out the DNA, mixed it with amphibian DNA because it was kind of fragmented, broken down, and recreated dinosaurs. Now you might think that that was a very fictional narrative, but as you might have seen, this year's Nobel Prize went to Pablo, and he does paleo anthropology, and what that exactly means is he goes into fossils of humans, extracts the DNA, and reconstructs the genealogy of humans, right? So precisely, there is a whole field of paleogenomics which goes into kind of these kind of embers, this kind of mosquitoes, extracts the dried bloods, reconstructs how dinosaurs were, how they were classified, and all those things. Though recreating dinosaurs might still be a very fictional thing, but what it tries emphasizing here is the fact that these these molecules, the DNA once again is a polymer of nucleotide triphosphates. As you can see here, you have the nucleotide, you have the sugar, and you have the phosphates. Again, you have the nucleotide, nitrogenous base, you have the sugar, and then you have the phosphate. And it's a repeating unit of such kind of molecules. And you will immediately see that ATP, that is so important for us to survive and run and speak and kind of sense and feel, it has to be hydrolyzed within seconds, if not minutes, for us to be living, right? So you cannot take 1,000,000 years for you to be hydrolyzing DNA. So the fact that these kind of molecules survive in the absence of enzymes for such long speaks a lot about the kind of rate enhancements that enzymes bring about. Enzymes really, really accelerate the kind of reactions and make everything compatible with life. And another thing that kind of fascinated me, so this was something that fascinated me a lot when I first learned about this. Another thing that kind of fascinated me was the kind of time scales on which things operate on a biological scale. You can see that they can operate on a time scale of 10 to the power minus 15 seconds. So I mean, 10 to the power minus 15 of a second. Can you believe you have bond vibrations happening? If the bonds do not vibrate at that particular time frames, then it's kind of going to create trouble. Likewise, for an electron transfer reaction to happen, it takes 10 to the power minus 12 to 10 to the power minus 11 seconds. And then you have the loop motions in proteins that we target for drugs. And the time scales for such loop movements is 10 to the power minus 8. And then if you talk about a lot of things like substrate transport, catalysis, and all these things, you will see that all of them happen on the time scales of nanoseconds to microseconds, as shown here. So we need to really understand things at the time scales. Now, just to give you a more realistic example, I'm showing you here the biomechanics of how a baseball player kind of reacts to the ball reaching him. And you will see that from the time the ball kind of leaves the hands of this person right here, where it's clearly shown here. Again, these resources are from internet, and I have exclusively borrowed them for educational purposes here. So from the time it's released from here, and the time frame that it takes to travel to the batsman's position is approximately 125 milliseconds. So the decision to swing, so the batsman has to perceive the angle at which the ball is coming at him, process the information, because the eye will receive the signal, send it to the brain has to process it, then send it to neurons that will kind of control the muscles to position the bat at the right angle, and then swing his bat. And all of these decisions have to be made within the time frame of 125 seconds. And this shows how crucial it is that biological processes work on time scales which are sub-seconds rather than rather than minutes or hours or millions of years. They are absolutely incompatible with life. So this kind of fascinated me about pursuing biology, and that's precisely what I do in my current role. Try looking at processes that are happening on a millisecond to microsecond time scales. I also got fascinated by science because of the beauty that is depicted in this particular slide. And here I'm trying to reproduce a panel from the protein data banks, the huge repository of structure. And you can see that depending on the kind of proteins that you talk about, they are amazingly beautiful. They're complex, they are colloidal, they look weird by conventional metric of what we define as beauty. But then you will see a virus looks so beautiful. There is T3 or T5 symmetries. And I'm not going to go into details of all those things. And then you have these membranes, and then you have membrane proteins that are anchored into the membrane. And these are all the proteins that float around in your cytosol. These are antibodies that that protect you from foreign agents kind of infecting and bombarding you. And then there are all of these things that, like for example, aspartate transcarbamylase, and you will see that it has a beautiful triangular symmetry, threefold symmetry. You have six-fold symmetry for glutamine synthetase. So symmetry is everywhere for you to perceive and see, and it's amazingly beautiful. And the first time I got exposed to all these beautiful structures, I decided that I don't want to do anything except to pursue such kind of beauty as part of my profession. Now, coming to drug discovery is a very complex process whereby you start with a whole lot of different small molecules, and then you trickle down to one or two because of lot of things getting eliminated on the way, because you want to make sure that your drug molecule acts on your target of interest but doesn't cause undue toxicities in the process. And it's a lot of trials, a lot of collaboration across different disciplines like medicinal chemistry, computational sciences, enzymologists, people who are experts in expression and purification of proteins, people who are expert in devising assays to understand, people who are experts in setting up high throughput screens so that these millions of molecules can be screened against those few small targets. And then of course there are a lot more to it, because you have the bioscientists who tests it in cells and then subsequently takes it to animal models. So you need in vivo scientists who does that. And then all of this is done in a way whereby you kind of iterate with the chemists. So each round, so you make sure that it doesn't hit undesirable targets and cause toxicities that could be potentially lethal if administered to humans. So you need to make sure that you are kind of accounting for all these things before you say some small molecule as drug for the purposes of treating a particular disease conditions, right? Now, the way a small part of drug discovery where I kind of function is lead optimization. And what usually happens is you have a pocket on the protein surface, and then you try to make sure that a small molecule kind of fits purely there, and then kind of blocks the activity of the enzyme in case the activity of the enzyme causes undesirable disease-like side effects, right? So that's precisely the purpose. And then it's not so clear as to how this can be achieved, but then this is achieved by a lot of trials and errors working with chemists, where you try going back. And the fact is, unlike a lock or a key, which are kind of rigid entities, as you will see here, proteins are kind of very flexible. They're breathing, they're kind of, you can see that molecules are kind of in constant motion, and hence it's just an analogy, and I would request you to take it as an analogy. It's more like a hand in a glove rather than a key in a lock hole. So it's more kind of more flexible, more breathing kind of entities that you want to design and block the activity of. And it's a very intricate tango that you dance with the chemists, and a single mistake by you or a single mistake by them can take you on a track that is completely orthogonal to where you would like to go eventually. So what happens is there are structural biologists who solve the structure of the small molecule with the protein of interest. There are enzymologists who look at what happens to the activity of the enzyme in the presence of the inhibitor, and so on and so forth. I'm not going to go into details, but this is precisely the kind of excitement that I am exposed to on a day-to-day basis. With this, I would try concluding my message here and say that to be a STEM professional means to be inquisitive and passionate. And also, to be a STEM professional need not mean being stereotyped as a nerd, as a nerdy individual. Scientists comes in all sorts of shapes and colors, and I hope I could impress upon you that fact. And also, being a scientist can mean deriving inspirations and energy from diverse set of activities like sports, arts, and literature, a couple of them. I'm not a sporty person though. So these hobbies will kind of mutually reinforce your strengths and expand your perspective on how you apply the kind of knowledge that you learn from all these varied activities into the kind of intense investigation that you put into science. And being a scientist also means being a wanderer, as I think I amply demonstrated during the course of my talk. And on a practical level, there are multiple opportunities that are available to you as a STEM professional. Academic pursuits is just one of them. So you go ahead, finish your higher education, finish your PhD and postdoc, and then get to be a professor where you teach courses and pursue your own research. You can also take up very exciting industrial positions, especially with the kind of situations that we are facing on the drug discovery front. We need a lot of brilliant scientists within industries who can collaborate across different disciplines, speak each other's language, understand understand each other's language, kind of transplant their own skill sets into someone else's skill sets and come up with small molecules that could be potential therapeutic avenues to treat diseases. You can opt for editorial positions where you can convey the excitement of science that you feel through your writings, kind of judging others' writings and research. And you could be ambassadors of societies like I have been lucky enough to be, where you kind of speak to a lot of youngsters, speak to a lot of your own peer group, spreading the excitement of science that you feel. With this, I would stop and would take any questions that the audience has. Thank you very much.
Thank you, Bharath. That was wonderful. You can't see me. You don't know what's happened. There we go. Here I am. I look like a ghost then. That was great. I'm going to stop recording.